thank you for your great introduction. And I'm so happy and so honored to be here since I know that Rochester University is super famous for optics and optical imaging. I feel so excited to have this opportunity to present our recent work. So I changed a little bit about my seminar's title from Pico-Second laser scanning microscopy to metabolic nanoscopy in aging and diseases. So my talk will basically talk about two different directions. One is the technology development. Another one is how we can apply that or demonstrate that kind of technology in biomedicine. So while we image biomolecular and cellular dynamics, I think we all understand how important the metabolism can be. It is a process that maintains our life, and metabolism can include a lot of biological processes, which is critical for maintaining the living status, including the genetics and the metabolism. We are going to visualize these biochemical interactions in not only space, but also in time-lapse manner to understand the complexity of life. And this imaging can really have far-reaching implications for human health, as well as the scientific progress. So we have been involved in this program. It's an NIH program called HubbleMap. It's called a Human Biomolecular Atlas. So I'll give a laser. I think the laser will be helpful for later slides. So Human Biomolecular Atlas program is going to establish the global platform. It's open for all type of countries, people who are doing the biomedicine or human health. You can see those cells, how we can map those cell subtypes and to establish this library for references. And even for these people who are interested in disease detection, that they have a good reference, not only for brain, but also for internal guts. These are image examples that taken by our colleague in this HubbleMap program. You can see a lot of colorful protein molecules from different organ tissues, including intestines or even lymph nodes, and some are from tonsil or kidney or lung. But you can see that most of the molecules are from protein. So we can visualize protein easily, but the imaging technology for visualization of these fat molecules are very limited. We cannot easily target the fat molecule in the body. However, we understand that the lipids metabolism in the body are major events for maintaining energy homeostasis. So how the fat molecules are metabolized in the brain or muscle or even liver, or even in the guts, that's super important to understand. Along the human evolution, we observed an excessive amount of fat in the body, not only because of the lifestyle change, but also because of the diet. So we understand that extra amount of fat in the body can induce diseases, even cancer. So that's super important for us to understand the lipid metabolism and to quantitatively image them. So how? First, we want to talk about some metabolic imaging technologies in the hospital. We usually see PET or MRI. They are familiar technologies. We utilize them a lot. They can provide the metabolic activities from the body. However, the spatial resolution are low. So for biologists and medical researchers, they would love to see cellular or subcellular resolution of metabolic activity, because a single cell is a unit of life, and it's super essential to visualize the cellular machinery. And the optical image technology such as fluorescence or Raman can be a great tool to resolve that kind of spatial resolution. And I'm going to introduce the Raman-based technology. In the past, it was label-free, but now we introduced the bioorthogonal chemical bond imaging to visualize even the newly synthesized molecule, such as new protein, lipids, DNA, RNA, or carbohydrates, and distinguish that signal from the pre-existing older molecules. Then you can visualize the real metabolic dynamics in cellular level. As we are familiar with fluorescence microscopy, it's so widely utilized and so actually so convenient. You can purchase these fluorophore. They are commercially available, and then just target the protein molecules you want to visualize. And it's actually the Nobel Prize was in chemistry for the green fluorescence protein in the past. And my colleague Roger Chen also is one of them who won the Nobel Prize. The contrast of biomedical imaging can be, different types, even the spin of the nucleus or electron outside of the nucleus. But there's one thing that we are going to introduce is the motion between nucleus. Then we can image these chemical bond vibrational modes. That's the Raman microscopy that we are going to engineer it into a video rate imaging technology here. So that's a great contrast for visualization of chemical compositions or molecular compositions in cell or body or even in human organ. But the spontaneous Raman microscopy we are already familiar about. They provide these chemical bond vibrational information here. So lower right side, we can see different chemical bond really have generate different Raman spectrum profile. Simply, in theory, we can pick one of the Raman peak and image to map out the chemical bond spatial distribution. In reality, the sensitivity of spontaneous Raman microscopy is too low to do video rate image. But we know the advantage here, very good potential for imaging is the Raman spectrum is really narrow. So the peak are sharp and narrow. For fluorescence, they are broad and overlapping. Bleed-through signals can limit the numbers of molecules that you can image at once. So maybe 5 to 6 maximum right now, typically. And if you want to visualize 100 molecules, you need to do many different rounds of wash out, clean them, and stain them again, wash them, stain them, image them, wash them, stain them, image them to achieve a large number of molecules. But the Raman, because they have a narrow chemical bond peak, and you can select one peak towards maybe 50 or even more, easier to separate the signal from each other if you know the specific Raman peak for each molecule. So with that kind of potential, we also learned that each molecule, if you look at these Raman spectrum in our library, each molecule have their own Raman spectral profile. For example, we are familiar about cholesterol. Cholesterol molecule, they generate a specific Raman spectrum. If it's 100% purified cholesterol, we can see on top that there is a Raman spectrum for cholesterol. Omega-3, yes, you can measure omega-3 to generate another Raman spectrum. So I will consider this case similar to you have your own voice or you have your sound wave, I have my sound wave. So each person have their own sound wave. In the end, you can distinguish each person by analyzing the sound wave profile. So there are different molecules and different molecules have their own spectrum. That's the wave that we can distinguish the signal from each other and finally detect different molecules. As we mentioned that spontaneous Raman microscopy have low sensitivity, it's really hard to do video rate imaging. So in the past decades, people were working so hard to enhance the sensitivity or transition rate. Not until 14 years ago, when Dr. Sunny Xie and his fellows at Harvard University, they invented the SRS microscopy, which is called stimulated Raman scattering. It combines the Raman scattering and stimulated emission together to achieve a higher sensitivity. That means instead of using only one laser beam for spontaneous Raman microscopy, SRS utilizes 2 laser beams, the pump and the Stokes. When the energy differences between the pump and the Stokes laser beam matches the vibrational modes of the molecule of interest, then the sensitivity or the transition rate can be largely enhanced. So with the joint effort of 2 laser beams, you can actually have transition rate increase by 10 to the 8 magnitude. That can end up with a signal level of 1,000 or 10,000 figure. So with that kind of sensitivity, you can easily set up a microscopy to do video rate image. So the trick here is The Stokes beam side, we put a modulator because the energy differences between two laser beams are very, very small and a small portion of the energy differences. We want to remove all the laser noise from the laser side and achieve the high sensitivity here. Then by detecting the Raman loss on the pump beam side, we can achieve a video rate imaging for the chemical bond directly. So in theory, if we collect a Raman spectrum just with a focused laser beam on top of the sperm cell head, we know that the DNA molecule only deposits in the sperm head, not in the sperm tail. So if you focus the laser beam on the sperm head, you can generate a blue spectrum on the top left, and you can see the DNA backbone signal here, Raman peak of DNA here, 781, 1091. But if you move away the laser beam towards the sperm tail and only detect the Raman spectrum from the sperm tail, you collect a red color spectrum on the lower left side. So this red spectrum, you don't observe DNA signal anymore, but they are protein and lipids signal. Protein and lipids signal still existing here in the sperm tail. So in this way, you can see that if we want to image the DNA spatial distribution, simply pick up this peak 1091 to image, or pick up 781 to image the DNA. So how to achieve that video rate? In the lab, we integrate this SRS microscopy with different imaging modalities. So TPF here is 2-photon fluorescence. From title, it's MPF, it's multiphoton fluorescence. We can do 2 or 3 or 4 photon fluorescence, but basically in the normal daily application, we do 2-photon fluorescence a lot. Second harmonic generation is integrated with the SRS microscopy. So with this microscopy, you can just put the sample under the objective lens and collect a 2D or 3D image for these biological sample from even human biopsy sample. So human needle biopsy also can be done like that. So you can see different molecular information from different imaging modalities at once. It's automatically co-registered. So you don't need to worry about the localization. And here is one example from a human patient biopsy. A patient who had diabetic symptom. He was worried about the kidney. He wants to know if the kidney has some very little tiny changes on the molecular level. And we had the needle biopsy, less than 1 millimeter of tissue. For us, it's a lot of tissue. And we image it with 2-photon fluorescence on the left side, 2-photon fluorescence to see the NADH and the flavin to generate a redox ratio and changes, and the second harmonic generation in the yellow channel to see the collagen extracellular matrix, and the lower red color channel is SRS lipids. Also, we do the protein as well, other molecules you can also list, but I didn't list every single channels. I merged them together in different modality of signal. You can see a multiplex image to see glomeruli in the kidney, which is cyan color. They are the label-free signal detected by SRS. So these chemical bonds are mainly from protein from the glomeruli. Glomeruli is the structural information here. And the long purple color or magenta color, that's the vasculature system protein signal. They are also collected by label-free SRS chemical bond imaging. The red color are from the fat molecule in the tubule of these kidney. And the green color here is the second harmonic generation from the collagen fiber. So we can detect them. Morphological information are here, but you can analyze even more. For example, you can see subcellular resolution of molecular composition differences in different structural domain. Here is a glomeruli in green color. These are tubule side with different molecular composition. You really can see different signals, and it can be 3D. If you scan from top layer to the bottom layer, layers of the image can be reconstructed into a 3D volumetric way. So in this way, the structural functional interactions can be mapped out. So how we can interpret this image with the label-free imaging technology? It's multimodality. It's a multimodal image For magenta color, they are from second harmonic generation for the collagen extracellular matrix signal. And in the center round shape or spherical shape, here is the glomeruli. You can clearly see that the collagen fiber are deposited in the surrounding area in the tubule side, not inside the glomeruli. And the red color, these are protein composition signal from the cell type inside the glomeruli or tubule side. But the blue color, they are the protein composition signal from glomeruli, and also another green color, green color also inside the glomeruli. But you can also see the deposit of label-free chemical bond imaging from fat molecule on top, and with the green color on from the nerve. Nerve is also here. So you can see the structural information very clear at subcellular resolution, and the ducts also accumulate a lot of fat in the lower side. So these provide a lot of information, not only the morphological, but also the metabolic, maybe the functional interaction between different, for example, arteries, nerve, or collecting ducts, and do the quantitative analysis. So while it can be quantitative, SRS, you have the intensity for each pixel that is it has a linear relationship to those concentration of that chemical bond. So that can be really quantitative for analysis, layers by layers. This can be linked to the spatial omics. So we can integrate this technology. It's a label-free technology with the labeled spatial omics profiling together. Then that is really mapping out the metabolic activity, dynamics, and the function at single cell omics. So it's a real multimodal with codex together here. We actually doing the human lymph nodes to detect the disease development in early stage. So until now, it seems like a lot of interesting molecules can be already detected. We are not satisfied with these kind of information because we want to see more molecular information. For example, we really want to see not only the fat or protein differences. We want to see in the fat molecule, which is the cholesterol, which one is the omega-3, the subtype of fat. And also superresolution means we want to see subcellular organelle, for example, the mitochondria inside the cell, how the mitochondria metabolize, or how the lysosome, or even the small vesicles or lipid droplets inside the single cell looks like when they do metabolic changes. And the third one here, the challenge for us is how we can distinguish the newly synthesized molecule, and you can distinguish this signal from new molecule to from the old pre-existing molecules, and we can map out the real metabolic dynamics. So I will talk about one by one how we can select the molecular select different molecules by molecular specificity approach. The traditional method in mass spec give us a lot of inspiration. So it's similar that we understand the spectrum profile for each molecule. So first we establish the library to measure one by one. And you can establish, for example, a few hundreds of molecules in the library or even more. So after that, you know each molecule have their own Raman spectrum profile. Then you do the hyperspectrum image stack that provide spectral information on each pixel in the image. So each pixel you have that spectral information, and you know on that particular pixel there are many different types of molecules sitting there, but it cannot be unlimited. It is finite, and the concentration of these molecules can be very different. For example, the pixel number 1, perhaps you can detect the top five molecule with the high concentration, but the other molecule perhaps it's below the detection limitation, so you can just ignore. So the top concentration of these molecules can be actually measured by the reference matching with penalty algorithm. Here we defined, so the red color pixel you detect from a human brain image. These are identified as a high score sphingolipids pixels. So they have very, very high matching score for sphingolipids. High cholesterol is in the green color channel, and the blue color channel have higher triglycerides. If you merge them together, it's a label-free detection of different subtypes of fat molecule. So these are from the human brain tissue. And we can also apply this to actually do different lipid subtype detection for other types of tissue. This is a fat body tissue from Drosophila, and it's an organelle level. The cyan color here, they are organelle called the lysosome in the cell. So these lysosomes have a different molecular composition inside. We can first measure that lysosome's Raman spectrum profile and detect it. So you figure out all the pixels that have those similar spectrum are in the cyan color channel here. And these are triglycerides intensive pixels. So these are lipid droplets in the cell, in the fat tissue. So we can merge them together to generate those label-free detection for different subtypes of a molecule here. And my student recently just found that to enhance the molecular selectivity or chemical sensitivity, we can even do circular polarization to enhance this kind of detection. If you look at the right side, the linear polarization, if we image the protein molecule on top, usually protein and lipids have CH3 intensive, CH2 intensive chemical bond. So CH3 chemical bond is abundant in protein, but it also existing in lipids, the fat molecule as well. CH2 is abundant in fat molecule, and also you have some in the protein molecule. So under the linear polarization case, you can see those image showing the protein and the lipid signal quite equally. But if you put left circular polarization, you can observe very different image. So after the left circular polarization applied on the protein intensive tissue, you can see the protein signal still existing and it's maintaining the original status. But the image that you take from this CH2 chemical bond, which is abundant on the lipids molecule, you observe a huge differences. These places become dark void, right? So that means the lipid molecule interact with this light very differently. And you can see that decrease of the signal from CH2 chemical bond are much faster than CH3 chemical bond. So that's how interesting observation. We are also doing further experiments to confirm a lot of phenomena in other tissues as well. So if you don't know what molecule they have in the tissue, what we can do to distinguish different molecular composition. From this one, we did the unsupervised clustering. So in different tissue, if it's a mouse brain or human brain or Drosophila brain, whatever brain tissue you get, you just take the hyperspectral image and detect on all these pixels in the image to distinguish or cluster the chemical composition. You can say it's molecular composition, and automatically you can see different cell groups. In the human kidney is the same. And in the single cell that is doing mitosis, that means a single cell that is dividing on the lower right side. It's dividing, but not divided yet. At that moment, we take a snapshot. You can see the subcellular resolution of newly synthesized molecules. And you can predict perhaps the red color pixels here, it's likely newly synthesized DNA, right? Because the shape looks like a chromosome region of interest, but it's unsupervised, but you can actually confirm with other technologies such as Codex or fluorescence staining method to confirm these are what type of molecule. So as we just mentioned, the second challenge for super resolution, we want to see subcellular resolution. A single cell is the basic unit of life, and we really want to know the cellular machinery, how cell make new molecules, how cell utilize old molecules to generate energy. So that's the cellular machinery with organelle level activity. That's the mitochondria. And the mitochondria are basically from the mother's egg, right? We discussed during the lunch that our body have mitochondria that from our mother. So these are very interesting to learn how we can generate super resolution microscopy for SRS or for multiphoton fluorescence, because they are not single molecule microscopy at all. So for non-single molecule microscopy, we cannot use the traditional deconvolution approach at all because it's not useful. But we should borrow a Pointillism concept here. This painting is a very good example. We can see in a distance, the painting provides many features: woman, umbrella, dogs, children. But if you zoom into inside that picture, they are all dots. So it's a Pointillism concept. It's really good for our non-single molecule microscopy. That means you can actually precisely localize the massive amount of emitters if you constrain those emitters numbers and also each emitter's intensity, unit of intensity. And the question becomes, you want to find out to minimize the distance between the image and also the virtual emitter, which is this. So you want to calculate the minimize the distance between this real image and the virtual image. And then you can localize these emitters that you have already generated in the image and find out the precise localization will generate the super resolution image, such as a human retina image. Because the importance of human retina, everybody knows it's a part of the brain, right? Our eye retina is part of the brain. So it's like a window of our brain, and the chemical composition or lipids composition on these rod cells in the human retina on top here are extremely important. During the aging processes, we have observed a chemical bond change or maybe the molecular change here. Lipid subtype can be largely different. So how we can detect that layers of membranes, chemical bond composition change, we need the super resolution to identify that resolution here. So distinguish different layers of membrane on the retina is extremely important for study aging processes or other actually diseases in the human retina. In the subcellular resolution level, we can see the mitochondria in the magenta color here, and the lipid droplets is in yellow color here. Cyan color is those protein enriched organelle. So 3 different cellular organelle, they interact with each other. You can see that very clearly. And without super resolution, you cannot see them and morphologically even. And this one is not only morphological differences, also you can see the chemical composition differences between each organelles, like this lipid droplets. So some of the lipid droplets have more cholesterol. Some of the lipid droplets can have more triglycerides. So how we can distinguish that with super resolution and the chemical composition differences, we can really distinguish different lipid droplets in a single cell and groupize them in different colors. So I think at this moment, you really want to know, so how we can generate those new molecules in the cell or tissue or organ and distinguish that with from the pre-existing molecules. And this is DO-SRS. DO-SRS here is another modality on top of the 3 different modalities previously we just mentioned. The reason is because fluorescence probes are so big. If you look at the fluorescence probe, antibodies, they are a few nanometers, and quantum dots, they are 5 nanometers in diameter. Fluorescent protein, they are smaller, but they are still big. But organic fluorophores are even smaller. They are still bigger than the glucose or amino acids. So if you want to see glucose or amino acids, it's not ideal to label with fluorescence probe. These bulky probe is going to perturbate the function of small metabolites. So our idea here is to generate a single chemical bond. You can attach an atom and generate a specific chemical bond that allows you to track this metabolic activity of small metabolites such as amino acids or glucose or fatty acids or choline or acetate. And then you can see how they incorporate into different macromolecules. So this atom will be generated by an isotope for hydrogen is it's called deuterium here. It has extra neutron inside. So this atom is heavier than hydrogen. What happened when a heavier hydrogen is plugged into the newly synthesized molecule? D2O is the heavy water molecule. So deuterium can be ripped off and plugged into, they can be plugged into the small metabolites really fast. It's a millisecond level through the enzymatic incorporation. So after that, the small metabolites with the deuterium atom, they can be slowly incorporated into large macromolecules such as lipids, protein, DNA, RNA, carbohydrates. With the newly synthesized chemical bond, what happens in the Raman spectrum? Why it's so important to generate this chemical bond? So this question is going to answer, it's going to inspire us to think about the Raman spectrum measured from natural biological sample. No matter you measure this spectrum from plants or animal, no matter it's a mammalian cell or a plant cell, you can generate a spectrum pattern like this. It has peaks on the left side and right side, but no peak in the middle. This is from 1800 centimeter inversed to 28 centimeter inversed. It's called a cell silent region. So the cell silent region has nothing, no Raman peak until you generate a new peak or new chemical bond from carbon deuterium here. Then this carbon deuterium labeled molecule are newly synthesized. You can directly image this CD chemical bond to map out the newly synthesized molecules such as protein, lipids, DNA, RNA, carbohydrates in tissue or cell or human body. So this is one example. If a single cell is doing mitosis and we snapshot that moment, we can image these newly synthesized CD labeled. CD is carbon deuterium chemical bond. This is a blue color channel, and the green color channel is the CD labeled new lipids, and the magenta color is CD labeled new DNA. And this is three-color image can map simultaneously. You can see the three-color image of newly synthesized macromolecules. The reason is on the left side, although they are having the CD chemical bond in different macromolecules, they generate different Raman spectrum shift. So that's how we can utilize this imaging platform to see the lipids, new lipid synthesis in traumatic brain injury. When the brain is injured, the brain is doing healing processes by the brain cell. So these glia cells in the brain, they are synthesizing new lipids, a lot of new lipids. In the naive brain, which is not injured, we don't observe that kind of newly synthesized protein at all, lipids at all. So this is also great for tumor detection because tumors are dividing so fast. So they generate a lot of CD labeled new protein lipids molecule, and we can easily catch that intensity increase in these tumor cells. So these tumors are growing inside of the brain, and the surrounding area, they are brain tissues. The brain tissue doesn't have that kind of signal. However, the brain tumor cell have a lot of CD signals. So you can see how easily that the surgeon can detect these tumor cells and remove them. Because if they stay inside the brain, they will grow into a big tumor very, very fast. So this idea can be expanded to other metabolites such as glucose or amino acids or even choline fatty acids. If you attach a deuterium into on these small metabolites such as glucose. This is the glucose case. The glucose molecule have a CD chemical bond now. So when the mouse or the animal, you can be animal or human being, drink some glucose water, the glucose will be incorporated into different macromolecules such as top green box is glycogen, the purple box is DNA, RNA, and the orange box is lipids, the red box is protein. So different macromolecules that are derived from glucose can be imaged here directly. CDL is new lipids. CDG is a new glycogen in the liver. and CDP. In the same piece of liver, you see new protein in the red color channel. So 3 color channels, you can see the morphological differences between each other. The lipid is a round shape. The glycogen is inside of these hepatocytes, and the protein is more homogeneous. So this is really good to visualize these metabolic activities. If you attach the deuterium in another, for example, amino acids like glutamine, so we can see, and the hypoxia, the glutamine can generate different signals, also acetate molecule as well. There are hypoxia-induced lipids synthesis. So hypoxia, low oxygen really change our metabolic activity in the body. So it can be 3D, and you can do time-lapse imaging to see which cell, how much of the new lipids are synthesized in the body or in a live animal. So the live animal, if the live animal is dancing here, you image them with a video recording. It's like a video recording period. In a period of time, you can see where is the newly synthesized lipids deposit in the head or in the tail. So that can be really clearly showing at subcellular resolution with this technology. Even in single lipid droplets, you can distinguish the newly synthesized lipids domain and compare with those older lipids. If you let the animal drink more glucose water, it's like you let them eat more sugar, they have more newly synthesized lipids in these lipid droplets. So that means we can really target a specific cell in the body. So that is a fluorescence signal for specific subtype of a glia cell. Then we can see the new lipids turnover rate on that particular glia cell. And this is really precise to study even the Alzheimer's disease. So how we can study this for Alzheimer's disease when you do image? First, we want to express those specific group of glia cell with the magenta color fluorescence protein. So these fluorescence protein expression are only on a group of glia cell. So you can target that glia cell when the tau protein are overexpressed in the brain. That means these brain are going to be Alzheimer's brain. So at that moment, you can visualize the newly synthesized lipid droplets accumulated in these tau overexpressed brain. And that's really detecting the metabolic changes in the Alzheimer's disease development. So during that process, we can rescue, how you gonna remove the Alzheimer's disease. You can remove the Alzheimer's disease by changing the lipids or fat molecules metabolic pathway. So by activation of AMPK metabolic pathway, you really can rescue that phenomenon to reduce the lipids deposit and also recover or maybe rescue that the Alzheimer's disease symptom. So there are many things that we can do about the metabolic imaging platform, using the metabolic imaging platform, such as diet regulation. So this is the final application I want to show you. If we eat less, we live longer. So this is a lifespan. DR is diet restriction. When we eat less, we live longer. These are animal studies, not human studies yet, but animals really live longer when they eat less. But the question here is, what happened metabolically when they eat less? What happened in the brain or other parts of the body? So we look into the brain, we can observe when the female become old, they eat less, they can actually elevate the metabolic turnover rate for these brain lipid droplets in the brain. The lipids synthesis is elevated compared with the normal food. But for the male, they also elevate the lipid droplet synthesis. So what is different from male and female? When the male eat more sugar, they still maintain the high metabolic activity, but when female eat more sugar, they reduce the metabolic turnover rate in the brain lipid droplet. That means the female brain perhaps function badly when they eat too much sugar, but the male still maintain strong metabolism and they're relatively okay. So to summarize my talk, I think 3 imaging technology that we mentioned. PRMSS is to detect the diversity of the biomolecular biomolecules in cell and tissue. A part is for super resolution to see subcellular organelle like mitochondria or lysosome or lipid droplets. Bioorthogonal is for newly synthesized molecule. So we can combine all of the three technology together to see subcellular organelle level, super resolution, metabolic dynamics, and you can study this for aging or disease-related diseases. Thank you so much for your attention. I would like to acknowledge my group members at UC San Diego and the collaborators at UC San Diego or outside of UC San Diego, and my funding source as well. Thank you.
Thank you. Well, I learned a lot about imaging today. I feel like, but anyway, let me see if there are any questions in the audience here.
I'll start if I can. So spectra usually are affected by the environment. I don't know about Raman spectra, but are there homogeneous or inhomogeneous broadening mechanisms that you have to be aware of when you're comparing different these different spectra, these different molecules?
Yes, environmental change can affect the Raman spectrum. Yes, so the spectrum shape, peak position, 4 with half maximum, we observe those each peak in the Raman spectrum could be detected differently under different chemical environments. So, for example, when we induce the protein molecule to change their secondary structure, such as alpha helix or beta sheet in the protein molecule, you really observe in the amide region located around 1655 centimeter inverse, they have a peak shape differences or peak position shift. So that can be observed, and also manipulating the pH value, they also change a little bit Raman shift temperature as well. So that can be studied in detail. Some PhD students, they are interested in that. So they actually manipulate the ion concentration as well to generate different peak spectral information and do analysis on that.
Let me ask one question. So in your work, you're mostly looking at Raman scattering for imaging. Do you use any other kind of fluorescence?
Yeah,
both of them?
3 of them actually. The second harmonic generation are also utilized. Yeah.
So the Raman has the advantage. First of all, you said there was a lot more. You stimulate Raman scattering on the spontaneous, you get much more signal. I mean, it's a much more intense signal you can get. And also resolution is better, right? You think resolution is better because I understood they compare to other techniques. The Raman is better for resolution. Is that
so the resolution is enhanced by the A-pod algorithm. After the image, we can use the A-pod to enhance the resolution.
I see.
Yeah.
I see. Okay.
Yes.
That's what you're doing.
Yes, yes. The spatial resolution, but the spectral resolution, we have 2 different resolution, right? The spectral resolution or the spatial resolution.
Any other questions here?
Yes.
So do you do all of this seems to be on the Stokes side? Do you do anything on the anti-Stokes side for Raman?
No, I didn't do that anti-Stokes yet, but it's very interesting to observe any signal differences. We are doing the Raman loss on the pump beam side. So we can also detect the Raman gain in the Stokes beam side. This is a good project for my PhD to do. So he's trying to do that by detecting the Stokes Raman gain signal. So we have 2 laser being collimated and coherent. And after penetrate through the tissue, they have the energy loss. Maybe you can say Raman loss on the pump beam and the Raman gain in the Stokes beam side. So by detecting each, or maybe either of that laser beam, you can generate, theoretically, you can generate the same signal, right? But in reality, you really want to compare which one is a stronger signal that you can detect.
Yeah.
It's a good question.
And one more question is, most of your work is not in vivo, right? You're not doing in vivo imaging, you're doing in vitro, I guess.
Exactly. The image that I show, most of them are ex vivo or in ex vivo or in vitro.
Oh,
Yeah.
Yeah, right.
Yes.
Right.
Yes, but you can do. So I showed one image that animal is dancing, right?
Right.
The small animal is dancing. That's in vivo.
Okay, I see.
Yes. So you can do in vivo video recording to see the animal is synthesizing new lipids or protein, and localize where is the newly synthesized protein or lipids molecule in the body.
I see.
Okay. Yeah, it's a great question. Yes.
Could you use the deuterium carbon bond as like a label to see newly synthesized molecules? I was wondering how long those molecules then exist in the cell before they start to like diminish. Like, can you use it to track like how long certain molecules persist in a cell before they're replaced?
Yeah, it's a great question. So you actually can do pulse chase. So how fast it can pause? Pause the signal. It's the first hand signal when you attach the deuterium on the newly synthesized molecule. How fast it can be. The C. elegans, which is a dancing animal that I showed, really fast. It's 20 minutes. So after 20 minutes, you can see substantial amount of CD labeled new lipids molecule in the animal. But in human brain, it takes a few days. And the glioblastoma I showed you in the mouse brain, when you grow a tumor inside the mouse brain, that mouse brain, you can perhaps label 1 day to see the signal. So it depends on the metabolic activity for each organ tissue. And the skin cells are very fast. It has sebaceous glands. People who have acne on the face, you are concerning about the sebaceous glands, how they metabolize lipids, because they generate lipids to moisturize our hair and skin. So if the sebaceous glands have problem or they are having the inflammation, you can see the altered metabolic activity in a few hours. So it's fast. It's relatively fast, but not as fast as C. elegans small animal, which is 20 minutes. In cell, it's about 30 minutes. And the brain can be days.
Let me see if there are any question on the Zoom. Anyone has any question on the Zoom here? People who are on the Zoom here, you can either type in the chat or you can ask question directly. I don't see anyone raise the hand here.
Okay. Thank you.
Yeah.